everyone, in this video we will be seeing about cytochrome, their types, cytochrome C oxidase, cytochrome p450. These are all some of the enzymes that are responsible for very important body functions. Like during excretion, we need soluble compounds so that it could be excreted via the urine. So how these soluble components are made and how our body we are getting energy. So initially we will see about cytochrome. So this is found in plants and in animals. So their main function is to carry the electrons. So we will see the structure. So this is the metalloporphyrin part of this cytochrome. So this is quadplanar where the metal is the iron and it is attached to 4 nitrogen group. But this is octahedral. It means it has 2 axial ligands above and below the ring. Based on the type, the axial ligand will vary. We will be seeing about it. So, this axial group will be bound to a place in the protein. The place where it is bound to the axial ligand is called the apoenzyme. That is the place of the protein where this axial ligand is bound is called the apoenzyme. And then the whole enzyme, that is the protein part and the prosthetic group part, entirely it is called the haloenzyme. And then since Fe, that is iron, is the metal, we can call it as a heme group. Okay. Then there is octahedral arrangement in this case. So there are 3 types: cytochrome A, cytochrome B and cytochrome C. We can also tell them as heme A, heme B or heme C. In the case of heme A, the R1 is CH double bond CH2. So this is R1 and this is R2, and the R2 is C18H30OH. And in the case of heme B, R1 and R2 both are CH2, that is CH double bond CH2. So this is found in hemoglobin and myoglobin. So this is the substituent that is in hemoglobin, myoglobin, then peroxidase and all. And then heme C, where R1 and R2 is a thioether linkage. See, CH, CH3, yes, and it is also bound to a protein. So this is very important, you have to note. In the case of heme A and heme B, only the axial ligands are bound to the protein part. But in the case of heme C, there is 2 other linkages that is also bound to the protein part. So you might have learned about hemoglobin, where you know that one of the axial ligand is bound to the histidine group, whereas other is bound to a oxygen group in the case of oxyhemoglobin. There is also chances for carbon monoxide or cyanide group to bound there. But in the case of cytochrome, there is no possible for other things like carbon monoxide or cyanide to bind. Why is it that means? Because both the axial ligands are bound to the amino acid of a protein. There is no way others can come and attach to these axial positions. So in the case of eukaryotes, so what does eukaryotes means? They have a nucleus and a membrane bound organelle. But in the case of prokaryotes, there is no nucleus and a membrane bound organelle. So some of the examples of eukaryotes are animals, plants, fungi, protocells. And for prokaryotes, we can say bacteria, archaea. You can learn in more detail about eukaryotes and prokaryotes. In the case of eukaryotes, this cytochrome C structure looks like this. So tuna, so this is a eukaryote, and the structure of the cytochrome C is looking like this. So see, this is the porphyrin part and this is the histidine group, that is one of the axial ligand, and this is the methionine group, where this is a sulfur group. Sulfur is attached and then here nitrogen is attached. And we can also see in the case of cytochrome C, there are some other linkages with the protein. That is, as I have already told, it is a thioether linkage. This is only found in cytochrome C. But in the case of cytochrome B and A, both the axial ligands are only histidine groups. There is no other linkages with the protein and all. So how is the electron flowing? What is the order from what it is flowing means? Initially it is mainly based on the reduction potential. We know if a molecule or something is having high reduction potential, it will reduce well and it will not oxidize well. That is, oxidation means loss of electron. So here A is having, that is cytochrome A is having high reduction potential. So they may not be able to donate easily the electrons. That is because they are having a low oxidizing power. But in the case of B, this is having a low reduction potential, so it can donate electrons very easily. So initially the electron is donated from B to C and then to A and then finally to oxygen. This order is based on the reduction potential. How this reduction potential are varying for various cytochrome means? This is because due to the porphyrin substituents, that is the substituents, because each one is having different substituents, and there is also the changes in the proteins, their positions, and the changes in the axial ligands. These are also responsible for this reduction potential changes. When it is getting oxidized, that is when it is losing its electron, initially Fe will be in +2 oxidation state, not this, and then later on after losing its electron it will be in Fe3 plus state. So why is this electron transfer reactions means? Because we know during cellular respiration glucose is getting oxidized. That is, oxygen plus glucose will be converted into carbon dioxide, water and energy. So during this process energy is released. So these electron transfer processes will be helping in the gradual release of energy and also store that energy in the form of ATP, so that our body can use it whenever we require it. So during this electron transfer process, the cytochrome C oxidase is also playing a role. So we'll study in detail about cytochrome C oxidase and then we'll see these 3 points. So this cytochrome C oxidase has 2 metals, copper and iron metals. They are found in the mitochondrial membrane in the respiratory chain. So what is respiratory chain? This is where every process is happening. There is the production of energy and then the conversion into ATP. There are various enzyme complexes and mobile electron carriers responsible for these processes. These are all present in the respiratory chain. If you learn in more detail about respiratory chain, everything will be very easy to understand. So generally what is the cellular respiration process? Glucose is binding with the oxygen and converting into water, carbon dioxide and energy. So this cytochrome C oxidase will be responsible for converting that oxygen into water. That is, oxygen is gaining protons and then it is reduced to water. So this is catalyzed by cytochrome C oxidase. We will see how it is doing this process. So the structure of this cytochrome C oxidase is having a multi-subunit structure. So generally the structure is not revealed clearly. This is having these many parts and this is the overall structure. This is not clear in books. They haven't found it, so that they have just given these are all some of the parts, and using various characterization methods they have shown something to understand what is going on. So it has 2 copper and 2 heme centers, that is copper A, copper B, heme A or cytochrome A, heme A3 or cytochrome A3. So we can separate this into 2 categories. Electron transfer is happening in copper A and cytochrome A, whereas the oxygen binding is happening in copper B and heme A3. So electron transfer is happening in copper A and cytochrome A. How is this electron transfer is happening means? Initially, first we learned about cytochrome and their types, right? There we have seen cytochrome C. So once the cytochrome C is getting the electron, it will donate that electron to this copper A, and then this copper A will donate the electron to heme A, and then later on this heme A will be donating to the site where this oxygen is being reduced. So the binuclear site is formed by heme A3 and copper B. They will be acting as a pocket where this oxygen is binding, and then it is reduced into water. So that this heme A and copper B is responsible for pumping H plus ion. So this also we have to note how we are getting H plus ions. For reduction, heme A and copper B is giving this H plus ions. So this H plus ion is provided by them through the entire mitochondrial membrane. So initially, before oxidation, the 2 copper will be in the plus 1 oxidation state, and the 2 Fe which I have mentioned will be in the plus 2 oxidation state. Later on, after oxidation, the copper is in the plus 2 oxidation state, and Fe is in the plus 3 oxidation state. So now we will go into the continuation of the process. Now we will know how this is going on. So what is happening is the cytochrome C that I have mentioned. So the cytochrome C will donate the electron to copper A. What is copper A? That is present in the cytochrome C oxidase, and then later on to the heme A, and finally it is given to the oxygen that is bound in the copper B heme A3 site. Okay, that's it. Then we will be seeing about cytochrome p450. So this is also an enzyme that is responsible for producing soluble compounds, that is it will convert insoluble one into soluble one. So that is their function. So this is inserting oxygen. So we'll see from the first. So this is found in plants, animals and in bacteria. They have found like 100 to 200 different p450 enzymes are present in human. And then in cytochrome p450, iron is the metal. So from where does it got this name p450? So P stands for pigment, and this 450 represents their solid band that was observed in the spectroscopic studies, and this was observed at 450 nanometer. Why means this band, that is the solid band, is due to the pi to pi star transition, that is blue to red transition, that is red shift, bathochromic shift, shifting to higher wavelength. So because of this band is produced, and then it is observed at 450 nanometer. That's why it got its name P450. So what is their function means? So it is a monooxygenase. Generally oxygenase means enzyme that is inserting an oxygen into a substrate. If it is a monooxygenase means it will insert only one oxygen atom into a substrate. So that where is this oxygen coming from to insert it means it will be coming from the O2 atoms. That is, one of this oxygen from the O2 molecule is inserted into the substrate, and the other oxygen which is left will be reduced, that is it will gain proton and it will be converted into H2O. So this we will be seeing with the overall very superb mechanism. So these are all some of the process. NO3 is converted into O3 and O2S is converted into O2SO. As you can see, one oxygen is added, and then conversion of hydrocarbon OH to ROH. RH. So this is the mechanism. So these are all the various things used in the mechanism. We'll be seeing in detail. Oxygen to H plus to electron, and then it is converted into ROH, which is a soluble form. and then conversion of aldehyde to carboxylic acid, alkene to epoxide. So these are all various other mechanisms, and these are various reactions happening in our body. Benzene is converted into phenol using this. Cyclone is converted into cyclohexanol. Then cyclohexene is converted into cyclohexene oxide. So you can refer it how oxygen is binding, and you can try to predict the mechanisms for various things. So we'll be seeing about the mechanism of how RH is converted into ROH in detail. So first we'll see the structure of this cytochrome p450. So this is the structure. As you can see, this is the metal porphyrin part, and so the metal is the iron. So of course we can tell it is a heme group because this porphyrin and the metal is the iron. So this is like a heme group. And then we can see one of the axial ligand is from the cysteine. Cysteine sulfur is attached, and the other is water. So these are all the two axial ligands. So this will oxidize insoluble hydrocarbons to water soluble compounds so that they can be excreted via the urine. So this Fe is initially in +3 oxidation state, and they are in low spin, and they are having an octahedral geometry. So this is also a simplified structure. You can see 5th site is attached to cysteine, and the 6th site is attached to the water molecule. So this is the mechanism. So I'll clearly explain with various points. If I explain here, it won't be that clear because the oxidation states are not clear here. So first what happens is, so this is our molecule, that is cytochrome p450. It is having Fe and then porphyrin P. So here P will be there. Okay. And then water is attached in the 6th site, and cysteine is attached in the 5th site. So here we can see that P represents the porphyrin part. So P represents this porphyrin part. Okay, this entire porphyrin part is represented as P, and this water and cysteine group is represented. Okay. First step, RH is added. So this RH will not be bound with the iron. It will bind in the protein part. So it is surrounded by the protein part, right? In the protein part, there will be some hydrophobic protein part. There this will go and attach, because since this is a non-polar group, there it can only attract well with hydrophobic part. So this will go and attach with some hydrophobic part that is present in our protein, and this is also present near this Fe center. So this will look something like this. So this is the Fe center, and water is gone. After RH is binding, water will be expelled. The entire place is now water free, and this RH is not bound in this side, but they are bound in the protein part. Now what is this Fe means? It is still in their ferric state, that is Fe3 state, but now it is having only 5 coordination. Initially it is in the ferric state, but with 6 coordination. After addition of RH, now it is only having 5 coordination. Later on, step 2. So now there is addition of 1 electron, that is reduction. So since it has got 1 electron, Fe3 plus will be converted into Fe2 plus. 3 is gaining, Fe3 is gaining 1 electron, and then it is converting into Fe2 plus. Here Fe2, that is the ferrous, is in high spin state. So initially it is in low spin state. After it is getting from the 6 coordination, it is converting into 5 coordination. While it is in 5 coordination, it is still in ferric state, but with high spin. Later on it is accepting an electron and again converting into Fe2 plus, and it is having a high spin state. So third step is oxygen is adding here. While oxygen is adding, they can be in two forms. If you have learnt about hemoglobin, you can know this. They are in resonance. We cannot tell they are in Fe2 plus state or they are in Fe3 plus state. We cannot predict. They may be in either of the state. Mostly they are in the superoxide form. So that next step, that is the fourth step, is addition of another electron. As you can see here, third step is over now. The fourth step is addition of another electron. Already they have one electron extra. Another electron extra will convert it into superoxide form. So that will make a bond here. So this is the structure now. There is a bond here now. So this is in their Fe3 plus ferric state. So what is step 5 is this oxygen is getting cleaved. So how is it getting cleaved means we are adding H plus, that is if you are adding 2 H plus to it, what will happen? They are getting cleaved and then it is forming water. So this will be the formal oxidation state now. Fe3, oxygen will be having 0 state, and then this Fe3 will donate 1 electron and it will be converted into O minus, and then Fe5 will be formed as it has donated another electron. So how you can explain this means these two bonds are present. So these two bonds will take away this electron with this oxygen. Okay, imagine these two bond is made up of two electrons. Here 2 electrons and here 2 electrons. So this is taken up by this oxygen. And since it has got 2 electrons, 4 electrons totally extra, using that 4 electrons it can bind with H plus. Since H plus is not at all containing electron, it can bond with that 4 electrons with 2 H plus. Later on, now our oxygen is having only 1, 2, 3, 4 and this bond is having 2 electrons, totally 6 electrons. So they are not in their octet form, right? Now they need electrons to make them very stable. So that our Fe will be donating electron to this oxygen. That's what I have represented here. Initially it is donating 1 electron and then it is donating 2 electrons. So totally Fe has donated 2 electrons and it is now O will be in O2 minus. We can also represent it like O2 minus, or this 2 extra electrons will make a bond like this and this will be looking like a double bond. I hope you understand how this is working. So what happens is we are adding 2 H plus ions. So this is the main thing. We are adding 2 H plus ions. So what happens is initially the electron count will be looking like this, and this bond is also taking the electrons here, and this bond is also this bond electron is also taken by this oxygen. So now they have more electrons and then there is 4 electrons extra. Using that it will make bond with this 2 H plus. So now this oxygen is left with only 6 electrons, 1, 2, 3, 4 and 5, 6. So it will need 8 electrons to make them completely octet, so that it will gain that electron from the Fe. So Fe is donating 2 electrons now and is converting into Fe5. I hope you understood it. Later on what happens is there is some cation radical is also formed. So we can see this porphyrin structure will be having this double bond in it, and this is it donates one of its electrons. So from this place it has donated the electron, so that place is represented positive since there is a loss of electron, and this electron will be free and they will be having like a radical. So this is called cation radical, and to whom they are donating this electron means they are donating the electron to the Fe. So this form can also be represented as P dot Fe4 double bond O. What does this mean means This is P means porphyrin. Okay, P means porphyrin. This dot represents the radical that is present in the porphyrin. So this is the radical that is present in the porphyrin, and it has donated one electron to Fe. That's why Fe is gained electron and is converted from 5 to 4, and then double bond O. So that then P dot means porphyrin radical cation. So I hope you understood it clearly. So we can represent it like this or this, and this can also be represented like this because of some of the radical mechanism going on. Then final step is CH bond is broken homolytically. So we will be seeing the mechanism of this reaction. So what happens is now OH is present and Fe5 double bond O. We have took this as how it is finally present. That is, this is the state. Okay, Fe5 double bond O, and this OH is present in our protein. What happens is initially this RH bond, this bond is made up of 2 electrons. So one electron is taken by OR and other is took by hydrogen. So now OR is having 1 electron and this hydrogen is having 1 electron. Then there is this bond between Fe and O. So this first bond, 1 electron is took by Fe and other by oxygen. So this is the extra electron present, and Fe took 1 electron and it is converted into Fe4. Since it has gained electron, it has reduced into Fe4. Later on what happens is this electron and this electron will make a bond, and now the structure look like this. Okay, then what happens is, so this is R dot, I should have put it upside, sorry. So now what happens is, again there is this bond between Fe and O. One electron will be given to Fe and other electron is taken by our oxygen. And so since Fe is got 1 electron, it is further reduced to Fe3, and now oxygen is having 1 extra electron. So this extra electron in oxygen, and already our OR is having 1 extra electron, so they both will make a bond and it is converted into ROH plus Fe3 plus. So this is the overall mechanism how ROH is formed from RH. So we'll call this process as a rebound mechanism. Finally, now OH is dissociated from the entire biomolecule and left via the urine wherever it has to be sent. So there is a shortcut for this process. That is, instead of adding oxygen and instead of adding the electrons, these steps can be bypassed by adding XOOH and peroxide. That I have mentioned here. See, instead of adding oxygen and instead of adding electrons and all, and instead of adding protons, see, these steps are bypassed by this short step. Okay, so if we follow this route directly, we will reach this Fe5 double bond O state, and then it will be like a small process, so that because we are reaching directly to the 5th step. So that's it about cytochrome p450, cytochrome C oxidase, and their various cytochromes types. So I hope you like this. So my references are Principles of Bioinorganic Chemistry by Stephen Lippert and Berg, and Organometallic and Bioinorganic Chemistry by Ajay Kumar. Thank you for watching my video. I hope you support me very well. If there is any doubts, you can mention in comments. Once again, thank you.